Feedback is one of those most basic mechanisms that control body systems. Positive feedback and negative feedback being the two major types. In positive feedback, the stimulus and the response are going to be going in the same direction, and in negative feedback, they're going to be going in opposite directions. This is going to control a wide variety of responses like thermoregulation and osmoregulation, but with the feedback systems, it functions kind of like a room temperature thermostat. That would be an example of a negative feedback loop. As the temperature goes up, you stop producing heat, and the temperature goes down. Animals can be regulators or conformers for particular environmental variables. So in this example, we have river otters. They are temperature regulators, whereas largemouth bass are temperature conformers. Endothermic animals like birds and mammals are going to use metabolic heat to regulate their body temperature, whereas the ectothermic animals like snakes and most fishes are going to gain most of their heat from external environments. Animals that are mainly endothermic, they're going to be sort of this broader sort of so-called warm-blooded structure. But we need to realize that endotherm and ectotherm are not sort of mutually exclusive strategies. So organisms can use behavioral systems to absorb heat or release heat, like radiation, evaporation, convection, and conduction. Heat is always going to be transferred from a hotter object to a cooler object. So the essence of thermoregulation is maintaining rates of heat gain that equal rates of heat loss. Because their heat source is primarily the external environment rather than the internal environment, ectothermic organisms are going to require much less food than endotherms of equivalent size, whereas the ectotherms are extremely effective and successful strategies at dealing with widely varied environments. So behavior or nature nurture or evolutionary success, all of these things are going to tie together into the broader survival mechanisms of heat balance. Some more specific examples might be the dense feathers providing insulation in a penguin, which you can see a layer of on the left, or the countercurrent exchange system of the veins and the legs of the Canada goose, allowing for efficient heat exchange and transfer between this flow. If you look at the human brain, for example, you have the hypothalamus. That is going to be acting like the thermostat. A group of nerve cells in the brain work together and they detect temperature changes. So if the body temperature is going up, you're going to sweat, your vessels are going to dilate, your body temperature is going to decrease as heat loss goes up. But if the temperature goes down, you're going to have shivering and constriction of blood vessels, causing body temperature to go up. But ultimately, you're looking at that balance of heat loss and heat gain. When you're looking at this sort of sensory response, this detection of stimuli, that really gets down to cell signaling, because all of life functions can be reduced to the cellular level. Multicellular organisms release signaling molecules that target cells, and those can then initiate a response. Some transmitting cells release local regulators that influence cells in the local vicinity, and you can really turn that into 4 types of signaling. You have the autocrine signaling, which is when a cell releases a signal that will act on the secreting cell itself, whereas juxtacrine is going to work on cells that are physically connected, that are going to be touching. So juxtacrine is going to be going something between the cells of a tissue, for example, whereas in paracrine signaling, it's going to act on cells that are near the secreting cell. A very specialized form of paracrine signaling is synaptic signaling, with the neurotransmitters being released across the synaptic cleft. It is near the cell, but it's from a specialized source, so it has its own name. This structure can then be related to more long-distance signaling, which really comes down to the endocrine or the hormonal signaling, which is going to allow hormones to travel in the bloodstream or to travel through fluids to maybe nearby organs or anywhere throughout the cell. Hormones and local regulators range widely in the molecular size and type, but the transmission of these signals is key for any cellular response. All of those feedback components come back when you start looking at how these things work together. Feedback regulation and coordination with the nervous system is incredibly common in endocrine signaling. So in a very simple endocrine pathway, endocrine cells respond directly to an internal or environmental stimuli by secreting a particular hormone. That hormone will then travel in the bloodstream or in body fluids to target cells, where it interacts with its specific receptors. That will then bring about a physiological response, which could reduce the stimuli or increase it. Some of these things will be negative feedback loops, like the release of secretin is going to then alter the flow of bicarbonate. So secretin is released when the pH goes down. Bicarbonate is going to neutralize acid, so the lowering of pH will lead to the increase of pH, negative feedback loops. So negative feedback loops are the most common for homeostatic mechanisms, but positive feedback loops do exist. Unlike the negative feedback, which dampens a stimuli, positive feedback reinforce, leading to an even greater response. And it's important to a variety of pathways, like those of oxytocin, for example. In oxytocin pathways, mammary glands secrete milk. Milk released in response to oxytocin leads to more suckling and therefore more stimulation. And activation of this pathway is sustained until the baby stops. So when mammals give birth, oxytocin induces target cells in the uterus to contract. So until the baby is released, contraction will increase. So positive feedback loops do exist, but they are going to accelerate these processes rather than balancing. A great example of a homeostatic balance is looking at blood sugar. This is going to particularly relate to people with diabetes because the pancreas has two major types of cells, the alpha cells and the beta cells. Insulin is going to be released by the beta cells, and another hormone called glucagon is going to be released by the alpha cells. So these hormones are antagonistic to each other. When one goes up, it's going to have the opposite impact than the other on blood sugar. So somebody with diabetes, for example, is not making enough insulin or not responding to insulin. So when the blood sugar goes up, that insulin is not effective. What normally happens is insulin causes the blood sugar to be stored as glycogen in the liver and the muscle cells, and the blood sugar would go down. Whereas glucagon would cause the liver to release blood sugar to make glucose go up. The structure sort of fails when you have diabetes. Type 1 diabetes, you don't produce enough insulin. Type 2 diabetes, you are insulin resistant. Now, what actually happens when a cell encounters a secreted signaling molecule is you're going to enter the stages of cell signaling. In reception, a chemical signal is going to bind to a cellular protein that is typically at the target cell surface, in this example, or could be found inside the cell if the ligand is a nonpolar one. That signal will then pass through a series of other molecules in a process called transduction. Transduction may be a single step, but more often is multiple steps, a series of molecules. Those are then called relay molecules. The molecular details of these pathways are very similar in lots of different organisms, showing that this likely evolved from a common ancestor. But eventually the relay molecules will reach whatever the end goal is for this process and will initiate a response. The transduced signal will trigger a specific cellular activity. What that activity is depends upon what is activating. Overall, these stages are going to help ensure that crucial activities occur in the right cells at the right time and in the proper coordination with other cells of the organism. Overall, there are really four types of receptors. The first major type is an intracellular receptor. If you look at the picture, the aldosterone in this case, which is a hormone, is able to diffuse through the phospholipid bilayer. And we can see that that is made from cholesterol and it is a lipid, so it makes sense that its receptor will be inside the cell. In this case, you're going to have the situation where that hormone will bind directly and then it can initiate a response as is. So there is no real relay process because the receptor and the protein found will bind together. Here's an example from an FRQ from a previous year. Estrogens are small hydrophobic lipid hormones that promote cell division and they passively diffuse. Describe one characteristic of the plasma membrane that allows estrogens to passively cross. They're lipid, so the lipid nature is not blocked. In a laboratory experiment, a researcher generates antibodies that bind to purified estrogen receptors extracted from cells. The researcher used the antibodies in an attempt to treat estrogen-dependent cancers, but find that the treatment is ineffective. Explain the ineffectiveness. The hormone functions internally. So when they find these antibodies and they put them on the outside, they're not able to get through the cell membrane because antibodies are not made of lipid, they're made of protein. So since the receptor is inside of the cell and the antibodies are outside of the cell, it is not going to be effective. Some intracellular receptors will be just in the cytoplasm. Some of them will be directly in the nucleus, but they will typically initiate a response directly, unlike the other three types of receptor. A gated ion channel is a type of membrane receptor that can act as a gate when the receptor changes shape. When a signal molecule binds as a ligand to the receptor protein, the gate opens to allow the flow of specific ions like sodium or calcium through a channel. Binding by a ligand changes the protein's shape and opens the channel, and when the ligand dissociates, the channel is going to close. So these ligand-gated channels are going to be opened kind of like a physical key opening a gate, allowing for that flow of ions. The ions then can initiate a cellular response. This is very common for things like sodium and potassium and chlorine and calcium and protons. So ion channels are going to be classified according to their subcellular location, with these things occurring on the outside of the cell, but also on the outside of many organelles. That dissociation will cause the gate to close. Now, some gated ion channels are instead voltage gated. This is something that's common, say, in the nervous system, where what opens the gate is an electrical charge rather than a physical molecule. But either way, gated ion channels will allow ions to flow only when the gate is open. The largest family of human cell surface receptors consist of the nearly 1,000 G protein coupled receptors. The structure of several of these receptors have been figured out over the past few years, and we realize that they are increasingly more common than we believed. A G protein linked receptor consists of a receptor protein associated with a G protein. A G protein is called a G protein because it utilizes GTP for its energy source. And the G protein can act as a GTPase, allowing the hydrolyzation of GTP to GDP. So once it is activated, it can then alter that GTP to have an energy exchange. G protein receptors are extremely widespread and diverse. Something like vision and smell in humans depend upon these, lots of components of embryonic development, but the similarities suggest that this system evolved very early on in life. But what's going to happen is that the signal molecule will bind, the GPCR will activate, the G protein will activate, the G protein will then move to what's called usually some sort of effector protein or activated enzyme, and that will then trigger the cellular response. When the G protein is done doing its job, it will hydrolyze the GTP to GDP, and it will fall back to the G protein coupled receptor. It never leaves the membrane. It just moves back and forth, initiating a response by the effector. Protein kinase receptors are another major category here, and they come in a variety of types, but it depends upon the amino acid that's used in their construction. So the example in this picture here uses tyrosine, so this would be called a tyrosine kinase receptor. These are especially effective when the cell needs to trigger several signal pathways at once. The tyrosine kinase receptor belongs to this major class of enzymatic activities, with kinase being the key idea. A kinase is any enzyme that catalyzes the transfer of phosphate groups. So you have these two kinase proteins that are inactive, and when a signal molecule binds to both of them, they will come together to form a dimer. Once tyrosine kinase receptor dimers are bonded together, they can then activate 10 or more different intracellular proteins simultaneously through each of those tyrosines in this case. These things are able to autophosphorylate, which means they're able to turn ATP and capture that phosphate group, and then each of those now charged and activated receptors can initiate a separate response. Abnormal functioning of tyrosine kinase receptors is associated with many cancers, for example, with excessive levels of the receptor tyrosine kinase called HER2 on breast cancer cells correlating with a poorer prognosis. In some clinical studies, treatments with drugs that target these receptors have improved patient survival rates by more than 1/3. So we're definitely interested in how these kinase receptors work. The transduction process is usually a cascade of molecules, maybe relay proteins, maybe kinase proteins, that are going to be transferring and relaying signals from receptors to target molecules in the cell. It's usually a multi-step pathway, which is going to allow for amplification, control, and multiplicity. Amplification, because one signal can then trigger multiple signals. Control, because there are so many steps, there are many different control points that can be activated, and multiplicity, because one thing can activate multiple different pathways, not necessarily just more of the same. So this phosphorylation cascade is going to be a series of proteins working together to pass that energy signal. It's going to start by some sort of activator, which will turn on a kinase. Kinases are able to phosphorylate, so the kinase will use ATP and activate the next molecule in the chain. That process will continue until the kinases are turned off. And kinases are turned off by protein phosphatases, which will dephosphorylate the kinase and inactivate it again. The phosphorylation-dephosphorylation system acts as a molecular switch in the cell, turning activities on and off as required. At any given moment, the activity of a protein regulated by phosphorylation depends upon this balance, with extracellular signaling molecules controlling the initial process. That is then going to move on until the cellular response is triggered. If these kinase pathways are not turned off, that can lead to lack of control mechanisms like cancer. If you then look at the proteins that are outside of kinase and protein phosphatases, there are a wide variety of other signaling molecules that can be activated. And you can see in the example here, you have like scaffold proteins and relay proteins and mediators and modulators. They all exist, but signaling proteins are these large intracellular signaling molecules that generally function by activating the next signaling protein. It could be regulated through direct means, it could be regulated through allosteric means, because they are protein in basis, but eventually the signaling proteins will trigger a cellular response. Most of the time, they're either going to be in two major categories, as you can see on the picture on the left. They're either going to affect an enzyme or protein that already exists, in this example, a cytoskeleton regulating protein or a metabolic enzyme. That will lead to a quick response, but one that is not long-term, whereas it could activate instead a transcription factor, which while slower in response, will be more long-term. Secondary messengers are non-protein molecules that are going to be continuing these processes and will work with the signaling proteins. CAMP is a great example of it. CAMP is cyclic adenosine monophosphate, is ATP with two phosphate groups removed and turned into a circle. So the ATP has transferred the phosphate groups, it's being used for its energy purposes, and the CAMP can trigger new kinases and new responses. Outside of CAMP, there are a variety of other secondary messengers, but they can really be classified into three categories based upon their nature: the hydrophobic ones, the hydrophilic ones, and the gases. Hydrophobic molecules are going to be water-insoluble ones that are going to be membrane associated and will diffuse from the plasma membrane and into the intermembrane space, where they can reach and regulate things within the cytoplasm. The hydrophilic ones are going to be water soluble like CAMP, and they're going to be located within the cytosol. Gases, like nitric oxide, for example, or carbon monoxide, they're small and typically uncharged or nonpolar, so they can diffuse through both. So the hydrophilic ones will specialize in working in the cytoplasm. Hydrophobic ones will be able to diffuse, so they can work inside of organelles, and the gases can work wherever because they are so small. We said response can really fall into two categories. You're either going to have an immediate response by altering an existing protein, or you'll have the more commonly thought of one where it will act as a transcription factor and will alter genetic function. So the phosphorylation cascade will continue until the kinases will activate a transcription factor. Transcription factors will either increase gene production or inhibit gene production. It depends upon what pathway is activated. These things will then alter the production of mRNA and will alter the production of future proteins. So the response really is, does it activate an existing protein, or does it alter the production of future proteins? And that's really the two major categories. What we see, though, is that hormones, since they are traveling through the bloodstream and through bodily fluids, they can act all over the body. And whether the response occurs in the nucleus or the cytoplasm, it's going to be fine-tuned at a variety of points. But because it's traveling throughout the entire body, we find that there are differences in response and differences in the effects. Epinephrine, for example, that fight or flight hormone, when it binds to cells in the liver, it's going to cause glycogen to break down, sugar's going to go up, that's going to provide energy for that fight or flight response. But muscle cells are going to respond very, very differently. The skeletal muscle blood cells are going to dilate. That's going to allow for an increased flow of blood and more likely to be able to escape or run away from that predator sensation. Whereas the smooth muscle cells that are in the intestines are going to contract and they're going to slow down the flow. So you're going to be focusing on running away rather than digesting, but it's all triggered by the same process. What's different is it's either the receptors that are different, in this case we have an alpha receptor and a beta receptor, or it's going to be a different series of internal proteins and secondary messengers that are activated, scaffolding proteins, for example. So the structures are going to cause these changes. So the same signal can initiate many different responses based on the type of tissue that it's impacting, which allows us to utilize the same proteins in more than one pathway and increase the efficiency of our communication process.